Hello viewers, this is Big Jack, the physics tutor. So this is biology, paper 2, 2023, internal. The question reads, Figure 1 shows changes which take place when a plant cell is placed in a solution of known concentration from stage 1 up to stage 3. So we have stage 1 up to stage 3. Now according to the arrows that we have here, we are starting from stage 1 up to stage 3. Then apart from that one, we also have structures like the cell wall, structure A and structure B. We have question 1 or question A. Identify the parts of the cells labeled A and B. So the part labeled A, it is called the chloroplast or the chlorophyll. And then as for structure B, it is called the vacuole or the cell sap. Now, what you need to know here is that the cell sap consists of sugar, salt, and water. Now, if you mix sugar, salt, and water, you are going to find that the solution that you are going to come up with, it is going to be highly concentrated or higher concentration. So, suppose this one here is the vacuole or the cell sap. The cell sap here, it is this structure here or this one here. So it contains sugar, salt, and water. So it is highly concentrated. Question B, Roman numeral 1. Using the changes shown in figure 1, explain what could have been the concentration of the solution in which the cell was placed compared to the cell sap. So we need to compare with to the cell sap. Now, what you need to know is that there are three types of solutions. We have the first one, which is called hypotonic solution. So hypotonic solution, it is the solution whose concentration is lower compared to the cell sap. So the solution which is hypotonic, it is going to have lower concentration compared to the cell sap here. Then the second one, we have isotonic solution. When you talk of isotonic solution, it is the solution whose concentration is equal to that of the cell sap or to that of inside a living cell, which means the cell sap and the isotonic solution, they are going to have the same concentrations. Thereby, the water molecules can enter the cell or leave the cell by osmosis. But when you talk of hypotonic solution, the water molecules are just going to enter the cell, from hypotonic into the cell, not from the cell into hypotonic, but from the hypotonic into the cell. And then the last one, we have hypertonic solution. So hyper means higher. So hypertonic solution, it is the solution whose concentration is higher to that of the cell sap, which means the water molecules are going to move from the cell sap or from the cell into the hypertonic solution. Now, according to the appearance of the cell from stage 1 up to stage 3, the cell membrane has been pulled out from the cell wall. Now, when the cell membrane has been pulled out from the cell wall, which means the cell was undergoing cell plasmolysis or plasmolysis in short. So when a plant cell undergoes plasmolysis, it is going to lose water by osmosis. Now for a plant cell to undergo plasmolysis, which means it was placed in hypertonic solution, right? Which means it was placed in hypertonic solution. Because when a plant cell has been placed in hypertonic solution, it is going to lose water by osmosis. And when it is placed in hypotonic solution, it is going to gain water by osmosis. So the explanation here, we are just going to say, the cell was placed in hypertonic solution and it lost water by osmosis. So the cell was placed in hypertonic solution and it lost water by osmosis. Now, when a plant cell loses water by osmosis, it is going to undergo what we call plasmolysis. Now, plasmolysis leads to dehydration. Now, when a plant becomes dehydrated, it is going to become wheat. So, when a plant becomes dehydrated, it is going to become wheat. Now, we have two types of wheat. We have permanent and temporal wheat. So, when a plant is permanently wheat, it is going to die. But if it is temporal wheat, it can be reversed into a normal plant cell by placing it in hypotonic solution or isotonic solution. What would be the overall effect of this process on the plant? So, as you can see, this one is just plasmolysis, the process of plasmolysis. Now, when a plant undergo plasmolysis, it is going to be dehydrated. Now, that dehydrated is going to lead to a plant becoming wheat, right? So, the effect here, the overall effect is just going to be dehydration. So, we can just say dehydration. Then, apart from dehydration, we can also say wheating. So, we can say wheating. So, this one is I and L. Wheating, like this. So, that one is the overall effect of the plant undergoing plasmolysis. It is going to become dehydrated and it is going to become wheating. Then D. Which of the three stages of the cell in figure 1 could be classified as turgidity state? So, when a cell gains water by osmosis, it is going to become turgidity, right? So, as you can see on this stage here, on the stages here, we have stage 1, which is the normal. Stage 2, a plant is losing water. And then stage 3, a plant undergoing cell plasmolysis or plasmolysis in short. So, we can say the plant which is in the turgidity state, it is the plant in stage 1. Why are we saying in stage 1? This is because stage 1, it is a normal plant cell. When a plant cell is normal, it is going to contain water in it. So the answer here is just going to be stage 1. That's the answer. E. Name 2 other processes which can make substances to move into and out of the cell. So the first one, we have diffusion or diffusion in short. So we have diffusion. Then the last one is going to be the active transport. So the last one, active transport. Question 2. Figure 2 shows processes of the fat of glucose produced during photosynthesis. The letters C to present these processes. So we have the process of photosynthesis. So usually photosynthesis takes place in plants where carbon dioxide is going to be reacted to water to form glucose plus oxygen. So now the arrow here is just showing that during the process of photosynthesis, one product is going to be produced here, and what that product is glucose, but we also have the other product like oxygen here, right? Now we have E, which is showing the energy, and H, which is showing nucleic acids, G, cellulose, F, starch, E, fats and O's, D, sucrose. So sucrose is just the form of carbohydrates in which it is transported in plants, or carbohydrates are transported in plants in form of sucrose. And then we have C, amino acids, the building blocks of proteins. Now we have question A, which is saying define the process represented by the letter I. So we have letter I. Letter I, energy here is released. Now the process which releases energy, it is respiration, which means they are just asking us to define what respiration is. So respiration is the process by which living organisms releases energy or obtain energy from the food they eat. The process by which living organisms obtain energy from the food they eat, or releases energy from the food they eat. Question B. Identify one process in figure 2 which represents a catabolic process. Roman numeral number 2, anabolic process. So catabolic and anabolic, these are the two types of metabolism. Metabolism is the sum total of all chemical reactions that occur in the body. And metabolism is controlled by enzymes. Now when you say kata means to break down, then ana means to build up. So catabolism is the process by which large molecules are broken down to form smaller molecules. Now during respiration, glucose is broken down to release energy, and carbon dioxide is going to be produced. So an example of a catabolic process, it is respiration. Then an example of anabolism. Since anabolism, it is the process by which small units are built up to form large molecules. So an example, it is photosynthesis. Since water and carbon dioxide are small molecules, those small molecules are going to be built up to form, are going to be combined to form up glucose, right? So an example of anabolic process, it is photosynthesis. So we have to choose here the letters. So as for a catabolic process, we are going to choose I. So we have I. Then as for the anabolic, we are going to choose what? C. Why are we choosing C? This is because here we are having the amino acids. And the amino acids are the building blocks of what? Proteins. So we have C. Then let's come to C. Question C. If this fate of glucose was in the human body, Roman numeral number 1, identify two processes from figure 2 which would take place. So we can say H can take place, glucose can be converted into nucleic acid. So we have the first process which can take place in the human body. So we have C can take place in the human body. Apart from C, we also have E can take place in the human body. E can take place, let me say I can take place in the human body, and also H can take place in the human body. So we have the first one, which is C. Then apart from C, we can choose E. Then if you don't want to choose E, you can choose I. It's up to you to choose. Or H you can choose. Roman numeral number 2. State the form in which the glucose would be stored. So the glucose in human body can be stored in form of glycogen, or it is stored in form of glycogen. That's the form in which glucose is stored. Now this glycogen is stored in the liver and in the muscles. Question D. Name two industrial applications of process I. Process I, it is respiration. So respiration is used in brewing and baking. So it is used in brewing and baking. Then the last one, it is used in the sewage treatment. Sewage treatment. Question 3. The table shows two blood groups, A, B, and O, and their properties. Question A, Roman numeral number 1. Complete the table by filling in the spaces provided. So we have the blood groups here, the antigens present, antibodies present, donates to, and receives from. We need to complete the table. So when it comes to the antigens are present in the cell membrane of the red blood cells. The antigens are present in the cell membrane of the red blood cells. Then when it comes to the antibodies, these antibodies are present in the blood plasma, right? So when it comes to the antigens present in AB, the antigens are represented by the alleles, which is capital letter I. AB, blood group AB contains two antigens. it contains antigen A, I with capital letter what? A. Then the antigen for blood group B is going to be capital letter I to the power B. So to complete this one here, we are just going to include capital letter I to the power A and capital letter I to the power B. These are the antigens present in blood group AB. Now when it comes to the antibodies, blood group AB does not contain the antibodies. The antibodies, they are present in blood plasma. So here we are just going to say none. The antibodies are not present in blood group AB. Then let's come to blood group O. Blood group O, it has none antigens. Then when it comes to the antibodies, it contains antibodies which is A and B. So just write A and B. Blood group O contains antibodies A and B. As for the antibodies, we don't use the RU, which is capital letter I. We just say A and B. That's all. Let's come to Roman numeral number 2. Explain what an antigen is. So an antigen, it is a gene present in the cell membrane of the red blood cell. So an antigen is a gene present in the cell membrane of the red blood cell. So an antigen or antigens, an antigen is a gene present in the cell membrane of the red blood cells. That is what an antigen is. Now, question B is saying, explain why a person with blood group AB can receive blood from any donor. So the reason why blood group AB can receive blood from any donor, this is because blood group AB does not have an antibody to attack the antigen of the donor, right? So we are saying blood group AB does not contain the antibodies to attack the antigen of the donor. So the donor, let's say we have blood group A. If blood group A is the donor here, you are going to find that since blood group AB does not contain the antibodies, which means the antibodies from blood group A are not going to affect, they are not going to attack the antibodies in blood group what? AB. Blood group AB does not contain the antibodies. So the answer here we are saying, this is because blood group AB does not contain the antibodies which can attack the antigens for the donor. Then apart from that one, we have question C, which is saying, determine the effect of blood from a donor with blood group AB being transfused into a recipient of blood group O. So blood group AB, it is a universal recipient, which means it can receive blood from different blood groups, but it can't donate blood to any blood group, right? So I'm saying blood group AB can receive blood from different blood groups, but it can't donate blood to any blood group. So here they are asking us the effect of a donor from blood group AB being transfused into a recipient of blood group O. So the effect here is just that it is going to lead to agglutination, right? It is going to lead to agglutination or clumping of blood. So when this one leads to agglutination, you are going to find that the red blood cells are going to be clumped together, right? As the result, you are going to find that the flow of blood is going to be reduced. Question D. Explain the importance of screening blood for the purpose of transfusion. So the first one, to prevent agglutination. To prevent agglutination. Then the last one, to prevent the infection. Suppose the person that you are getting blood is having HIV and AIDS. So if you haven't screened the blood, you can get HIV and AIDS or any blood disorders. Question 4. Figure 3 shows an example of an ecosystem. So we have the sunlight here, water, small fish, carnivorous fish, land, and plants. Question A. Identify 2 non-living parts of the ecosystem in figure 3. So the first thing here, we have the sunlight, which is this one here. Apart from the sunlight, we also have the land. Apart from the land, we also have water. These are non-living organisms or non-living parts. But plants, small fish, and carnivorous fish, these are examples of living organisms. B. From the features of the ecosystem in figure 3, name the type of ecosystem. So this type of ecosystem, it is called the aquatic ecosystem. Okay? It is called aquatic. So this type of ecosystem, it is called the aquatic ecosystem. So when you talk of aquatic, these are the just living organisms that live in water. So aquatic ecosystem. Or you can just say pond. Question C. Explain the role played by the following in the ecosystem in figure 3. We have Roman numeral number 1, small fish. So small fish, they play a role by reducing the number of plants here. They are going to eat the aquatic plants here. As you can see, this fish here, this small fish here is eating a plant. So they maintain the number of plants in water. And then the carnivorous fish. The carnivorous fish is going to maintain the number of small fish. Right? It is going to maintain the number of small fish. In other words, the carnivorous fish is going to eat small fish by maintaining the number. Question D. How does the sunlight in figure 3 affect the distribution of plants in the ecosystem? So you are going to find that they are the sunlight, they are the rate of photosynthesis. As the result, the plants are going to be many. But the lower the sunlight, the lower the distribution of what? Plants. So they are the sunlight, they are the distribution of plants. The lower the sunlight, the lower the distribution of plants. Question 5, which is the last one. A. Define the following terms used in genetics. Roman numeral number 1, recessive allele. So a recessive allele, it is just a allele which is overshadowed in the phenotype, right? So it is overshadowed in the phenotype by a dominant allele. So it is overshadowed in the phenotype by the dominant allele. Roman numeral number 2, phenotype. Phenotype is an outward expression of a living organism. An outward expression of a living organism. The way you are composed of outwards. If you are dark in complexion or if you are brown in complexion, that is your phenotype. Then when you talk of genotype, it is an internal appearance or genetic composition of a living organism. Question B. Jane has blood group O. So blood group O can only exist as homozygous recessive. So blood groups are represented by alleles, which is capital letter I. So this is how you represent blood group O. Blood group O can only exist as homozygous recessive. When you say homozygous recessive, homo means same, so you're going to have the same O here. And recessive means the allele which is overshadowed. Right. So blood group O, it is the universal donor. So blood group O can donate to any blood group. Jane has blood group O and has a child of blood group B. She accuses John of blood group AB to be the father of her child. With the help of a genetic diagram, determine the possible offspring to conclude whether or not John could be the father. To answer this question, what you need to know is that blood group O can only exist as homozygous recessive. So blood group O can only exist as homozygous recessive. So that is how we represent homozygous recessive. Then as for blood group A, blood group A can exist as homozygous dominant and heterozygous dominant. So since blood group O is recessive or can exist as homozygous recessive, which means blood group O is a universal donor. It can donate to any blood group, right? It can donate to blood group A and it can donate to blood group B. Then let's come to blood group B. Blood group B can also exist as homozygous dominant and can also exist as heterozygous dominant. Then as for blood group AB. Blood group AB, it exists as codominance. So when you say codominance, which means blood group A, this means that blood group A is dominant and also blood group B is dominant. That's codominance. Now, since we know that blood group O exists as homozygous recessive, so the first thing here we are going to start with the phenotype. So the phenotype, it is just the outward appearance of a living organism. So we have Jenny. So Jenny is having blood group O. So we have Jenny being closed with John. John having blood group AB. So the next stage, we need to have the genotype, which is the genetic appearance or the genetic composition of a living organism. Now, as for the genotypes or the genotype, these are represented by alleles. So remember, blood group O can only exist as homozygous recessive. So we have blood group O. Then when it comes to blood group B, this one here exists as codominance. So we have blood group AB, not blood group B. Then from there now, what follows next is splitting the gametes. So during the splitting of gametes, this one is going to be independent as blood group O. And this one here is also going to be independent as blood group O. This one, blood group A, as independent. And also blood group B as independent. Then what follows next is random assortment or random fertilization, where this one is going to be crossed with this one here. Therefore, we are going to have blood group A. Now, this blood group A is just going to be blood group A heterozygous dominant. Then the next one, we are going to have blood group B. Now, this blood group B is just going to be also heterozygous dominant. Then the next one, this one here, we are going to have blood group A. Now, this blood group A is just going to be heterozygous dominant. Then the last one, we are going to have blood group B. Now, this blood group B is going to be heterozygous dominant. Now, as you can see here, we don't have any child with blood group O, right? Because blood group O only exists as homozygous recessive. So here we don't have any child with blood group O. So this one we are going to call it as the first phylum, which is F1 genotype. So to conclude, we can just say this one is going to be blood group A, then this one is just going to be blood group B, then this one is just going to be blood group A, then the last one is going to have blood group B also. So here we don't have a child with blood group O. So which means John, so we can conclude that John was the father to the child. Why are we saying John was the father to the child? Because the child was having blood group B, and here at least we have 2 children with blood group what? B. So John was the father. So that was the last one. Thanks for watching.